Welcome back, everyone. Today, we're diving into something really, really cool. We're talking about the possibility of predicting heart attacks, like actually predicting them before they happen. I mean, how amazing would that be, right?
It would be a game changer for sure.
Exactly. And get this, the key might lie in these tiny things called microRNAs, or miRNAs for short. They're like little genetic messengers that could hold the secret to early detection. We're going deep into a study from Biology Direct that used machine learning to analyze these miRNAs. Pretty wild stuff.
It is. And what's really fascinating is the idea that these tiny molecules could be like an early warning system.
Okay, so hold on. Before we get ahead of ourselves, what exactly are miRNAs and how are they even connected to heart health? Break it down for us.
Right, right. Well, miRNAs are short, non-coding RNA sequences. They're like fine-tuners for gene expression. They're involved in a ton of different biological processes, and a lot of those processes directly impact your cardiovascular health. Things like angiogenesis, that's the growth of new blood vessels, cardiomyocyte growth, that's the growth of heart muscle cells, and even your heart rhythm. So, what makes miRNAs so fascinating is that the changes in their expression might happen even before you see any noticeable symptoms, or even before you see changes in the traditional biomarkers we look at.
So these miRNAs could be like the canary in the coal mine for our hearts, alerting us to problems before they even become serious. That's amazing.
Precisely. And that's really where this biology direct study comes in. They use machine learning, which is really powerful, to analyze miRNA expression data. And they wanted to see if they could accurately identify individuals who had experienced a heart attack just based on these miRNAs.
I'm always so fascinated by how researchers use these massive data sets. Where did they get the data for this particular study?
They got it from the Gene Expression Omnibus Database, also known as GEO. It's like publicly available, which is really great. And it's a repository that houses a vast collection of genetic information. It's a treasure trove.
A treasure trove, yeah, I can imagine. So, walk us through the study's methodology. What were the key steps they took? Like, how did they do this?
Okay, so first they used two separate data sets, one to train their machine learning models, and another independent data set to test the model's accuracy. This is really important because it helps make sure the models are generalizable and not just overfitting to a single data set.
Oh, that makes a lot of sense. It's like making sure a student can apply what they've learned to a new set of problems, not just is the practice test.
Exactly, exactly. So initially they identified 99 miRNAs that were differentially expressed between people who had heart attacks and those who didn't. Then they used a statistical method called LASSO. It stands for Least Absolute Shrinkage and Selection Operator, which is a mouthful. But basically, it helped them narrow down that list from 99 to a panel of 21 key miRNAs.
Okay, so they went from 99 suspects down to 21. What happened next?
Well, here's where things get a little tricky. Because of platform differences in how the miRNA data was collected, only 10 of those 21 miRNAs were actually present in the independent test dataset. Which is, it's a challenge in research, it happens, but it just shows how important it is to have standardized profiling platforms.
So even with those limitations, they ended up with 10 potential biomarkers for heart attacks. That's still pretty impressive. Okay, let's talk about the machine learning models now. What were the researchers actually trying to do with those?
Essentially, they were training algorithms to recognize patterns in the miRNA data that could distinguish between individuals who'd had an MI, a myocardial infarction, and those who hadn't.
So kind of like teaching a computer to spot those subtle signs of a heart attack that humans might miss.
Yes, exactly. And they use three different types of machine learning models: support vector machine, or SVM, gradient boosting, or GB, and XGBoost. XGBoost is like a more advanced form of gradient boosting. We don't need to get into the really complex details of how these models work. But the main thing to remember is that they're designed to find patterns in complex data.
Okay, so did they find any promising patterns in this case? Were these models actually successful at predicting heart attacks?
The results were actually really promising. The models showed good accuracy in differentiating between individuals with and without a history of MI. They even combined the predictions of the three individual models into what's called a hard voting ensemble, or HVE. So in this approach, the models kind of vote on the outcome, and this led to even better performance.
So it's like getting a second or third opinion, but from a super smart algorithm. What kind of accuracy did they achieve?
On the independent test set, the HVE achieved an accuracy of 86%, and an AUC, which stands for area under the curve, of 0.83. So it's not perfect, obviously, but it's a pretty significant step forward in early heart attack detection.
That's amazing, especially considering they were working with a limited number of miRNAs in that test data set. What do these findings mean for the future of heart attack diagnosis and treatment? This feels like a game changer, but I imagine there's still a lot more research to be done.
Oh, absolutely. This is just the tip of the iceberg, really. But it opens up so many exciting possibilities for personalized medicine and, proactive healthcare. Imagine a future where a routine blood test could analyze your mRNA profile and tell you if you're at increased risk of a heart attack.
That's incredible. So, what are the next steps? What needs to happen to take this research from the lab to the real world?
Well, definitely larger, more diverse studies are needed to validate these findings and to address those platform differences we talked about. Imagine what we could achieve with standardized profiling platforms and, even larger data sets.
It seems like we're really on the verge of something major here when it comes to heart health. What stood out to you from this research?
One of the things that I found really compelling is that 9 of the 10 miRNAs used in the final models already have established roles in heart health. So this isn't just like some random coincidence. It really strengthens the connection between these miRNAs and the development of heart attacks.
That's fascinating. It means these aren't just random molecules floating around. They're key players in the intricate dance of heart health. Are there any details about those 9 miRNAs that you found particularly interesting?
Well, yeah, each of these miRNAs has like its own unique story to tell about its role in heart health, like miR199B. This one's involved in the remodeling of heart tissue after an injury, while miR200A seems to have like protective effects against oxidative stress, which, that can damage heart cells.
So it's like this constant battle between forces that help and hurt the heart. And these miRNAs are like right there on the front lines. What about some of the other miRNAs?
Yeah. So there's the miR29 family. This group of miRNAs is involved in the development of fibrosis, which is the formation of scar tissue in the heart. And too much fibrosis can stiffen the heart and make it less efficient at pumping blood.
So it's a matter of balance. Like even processes that might be beneficial in some cases can become problematic if they go unchecked. Were there any other miRNAs involved in this sort of like double-edged sword scenario?
Yeah, there's one called miR375-3P. It's a really interesting one because it's been linked to a process called ferroptosis, which is a type of cell death specifically associated with iron overload. And it seems that miR375-3P might actually be involved in promoting ferroptosis in heart cells.
Wow, that sounds intense. So we've got some miRNAs promoting heart health, others contributing to damage, and some even playing both sides, huh? What about the rest of the miRNAs on this list?
Okay, so there's miR132-5P. This one is thought to be involved in several processes related to heart failure. It's been linked to the enlargement of the heart, fibrosis, and even cell death.
Sounds like this one is definitely not on the heart's nice list.
Nope. And then there's miR133A3P. This one has actually been shown to have some protective effects against ischemic injury. That's the kind of damage that's caused by a lack of blood flow to the heart, like you might see during a heart attack. And interestingly, it's also been linked to platelet function, which is important for blood clotting.
So this one's a bit of a hero, fighting for heart health on multiple fronts. Were there any other miRNAs with connections to platelet function?
Yeah, there's miR142-3P. It's interesting to see these connections between, like, seemingly disparate processes. And then, of course, you can't forget about the inflammatory response that happens after a heart attack. Both miR335P and miR34A5P are involved in that.
Right. Right. Inflammation is such a complex process. It's crucial for healing, but it can also cause further damage if it becomes chronic.
Exactly. And then, of course, we have that 10th unstudied miRNA, HA-SMIGME601.
Yeah.
It really makes you wonder what other secrets are hidden within our genome just waiting to be discovered.
It's like finding this mysterious artifact. We know it's important, but we need to unlock its secrets to understand its true value. It's incredible to think that this research was all based on analyzing whole blood samples. So they were capturing information from a variety of cell types, not just heart cells.
Oh, absolutely. That's one of the strengths of this study. It gives us a more holistic view of how the body responds to a heart attack, at the molecular level.
Mm-hmm.
We're not just looking at the heart in isolation. We're understanding how the entire system is affected.
It really highlights how interconnected our bodies are, and to think they were able to achieve these promising results even with the limitations of the data. Dealing with those platform differences and having a reduced number of miRNAs available in the test dataset, it makes you wonder what could be accomplished with standardized profiling and even larger datasets.
Imagine the insights we could gain with more robust data. We might be able to uncover even subtler patterns and develop incredibly accurate predictive models.
It's exciting to think about the possibilities. But how do we translate this from research into reality? How do we move from the lab to, like, the doctor's office?
Well, that's the million-dollar question. The next step is to validate these findings in larger clinical trials. We need to see if these miRNA biomarkers can reliably predict heart attacks in, diverse populations and real-world settings.
It makes sense. We need to be sure it works for everyone, not just a select group of people in a research study. And then, assuming the findings hold up in clinical trials, what are the practical applications?
Ideally, we'd want to have a simple blood test, something that could be incorporated into like routine checkups, like a cholesterol test, but for heart attack risk.
Yeah.
It's still a long way off, but this research is a huge step in the right direction.
It's a future that could transform how we approach heart health, shifting from, like, a reactive approach to a proactive one. Instead of waiting for something to go wrong, we could be taking steps to prevent it from happening in the first place.
Exactly. And with the power of machine learning and the insights we're gaining into miRNAs, that future might be closer than we think.
Okay, before we get too carried away looking into that future, let's step back for a second and talk about the 21 miRNAs that they initially narrowed it down to before the platform issues limited them to 10. I know we talked about the 9 that already have roles in heart health, but what about the other 12?
Well, of those 12, we actually know something about 11 of them already. They're known to be involved in various aspects of heart function, like cell growth, cell death, the response to stress. So their inclusion in that narrowed down list of 21 just further suggests that they could be playing a role in the development of a heart attack.
It's like each new piece of information we get adds another layer to this complex puzzle.
Exactly. It's this interconnected web of molecular interactions that makes research so fascinating. And the fact that there's still so much we don't know, that just makes it all the more exciting.
I'm really curious to know more about the potential of personalized medicine and how this research kind of fits into that bigger picture. We touched on it a bit earlier, but I think it's worth exploring further. What does a future of personalized healthcare look like, and how could these miRNA biomarkers play a role?
It's a future where, like, treatments and interventions are tailored to an individual's unique genetic makeup and risk factors. Instead of a one-size-fits-all approach, healthcare would become much more precise and proactive.
It's almost like having, like, a custom-made health plan based on your own specific blueprint. How could these miRNA biomarkers be used in that kind of context?
Imagine, someone goes for their annual checkup, right? And as part of their routine blood work, they have their miRNA profile analyzed. And if specific miRNAs associated with heart attack risk are elevated, that would be like an early warning sign.
It's almost like a personalized risk assessment tool. What would happen next?
Well, it could start a conversation between the patient and their doctor about lifestyle changes, like diet and exercise. It might even lead to preventative measures like starting a low dose of aspirin or other medications. Early intervention is key when it comes to heart health.
Right. It shifts the focus from reacting to a problem to, like, preventing it in the first place.
Exactly. And this isn't just limited to heart attacks either. The great thing about personalized medicine is that it can be applied to so many different conditions. Imagine using similar approaches to predict stroke, cancer, Alzheimer's disease.
It's a future where we're not just living longer, but living healthier for longer. It's exciting, but I imagine there are challenges too.
Right, of course. Implementing personalized medicine on a large scale is really complex. We need to consider the cost of these tests and make sure that everyone has access to them, regardless of their background or income.
And we can't forget about the ethical implications either. We're talking about accessing and interpreting really sensitive genetic information.
Absolutely. Privacy concerns, potential discrimination, the psychological impact of, knowing your risk for a serious condition. These are all things we have to think about carefully. As we move forward, it's so important to have open and honest conversations about the potential benefits and risks of personalized medicine. It's a powerful tool, and we need to use it responsibly.
This deep dive has been amazing. We've covered so much. From the tiny world of miRNAs to the power of machine learning and the potential of personalized medicine. It's clear that we're at a really exciting turning point in healthcare.
It's a future full of possibilities. The more we learn about these tiny molecules and how they influence our health, the closer we get to a world where diseases like heart attacks are no longer a matter of if, but when, and when becomes a choice we can make through proactive healthcare.
That's a powerful thought. Thank you so much for joining us on this incredible journey of discovery. Until next time, stay curious and keep on diving deep.